Ladies and gentlemen, welcome to the DNA double helix.
In discussing DNA replication, it is important to understand the structure of a double helix and a nucleotide.
Yes, sir. First, the structure of DNA is largely formed by hydrogen bonds.
The nucleotide consists of a deoxyribose sugar, a phosphate group, and a nitrogenous base.
Well said. The four nitrogenous bases in the nucleotide are guanine, cytosine, thymine, and adenine. And these are all referred to as complementary base pairings.
Adenine and guanine are known as purines, while thymine and cytosine are known as pyrimidines.
Nucleotides make up most of the double helix, while the phosphate and deoxyribose make up the backbone.
Another important feature of DNA structure is histone. It acts as a spool of thread for the DNA to wind around. Without histone, DNA would be loose and unorganized.
Histones also allow for DNA to be packaged into small units called nucleosomes.
When we establish DNA, we must talk about replication. This is, of course, the copying of DNA.
For replication to occur, a helicase separates two parental strands in the same fashion that a zipper does, exposing the base. They unzip in opposite directions, and this is known as anti-parallel.
The product of this zipper splitting is known as the replication fork.
After the double helix is unwound, it is used as a template for the copies.
The enzyme DNA polymerase attaches to the split strand and adds new nucleotides to form daughter strands in DNA.
DNA polymerase always moves in a 5 to 3 direction along the strand.
The DNA polymerase then forms a covalent bond between the phosphate group of the new nucleotide and the 3-oxygen atom of the deoxyribose.
This means that replication occurs on different sides of each side of the molecule.
One strand called the leading strand is assembled in the direction of the replication fork.
The other strand, also called the lagging strand, is assembled away from the replication fork and must be done in segments.
The enzyme DNA ligase is then used to put these segments together.
After the DNA polymerase is finished, there are two daughter strands, exact copies of the parental strands.
Most replication is known as semi-conservative because it creates two daughter strands in one new strand.
Lastly, there are two differences between prokaryotic and eukaryotic cells.
While prokaryotic replication occurs in the cytoplasm, eukaryotic replication occurs in the nucleus.
Secondly, while prokaryotic cells can only have 2 replication forms, eukaryotic cells can have hundreds.